Hey, biology students, welcome to this explainer. Today, we're diving straight into the incredibly complex biochemical gymnastics of DNA replication, and we're going to break it all down into a step-by-step factory line tutorial. If you've ever looked at a textbook diagram of a replication fork and felt totally overwhelmed by the sheer number of enzymes flying around everywhere, you are in the right place. Today, we're going to organize that chaos. Here is our roadmap for this tutorial. We'll start with the blueprint, unwind the double helix, build the leading and lagging strands, and finally run through proofreading and repair. All right, part 1, the blueprint. Let's lay down the basic rules of our molecular factory floor. Take a look at how these DNA strands are oriented. It's like the lanes of a highway, right? They run in completely opposite directions, which we call an anti-parallel arrangement. Up at one end, you've got the 5' end sporting a phosphate group, and down at the other end is the 3' end, which has an OH or hydroxyl group attached to the shooter. And here's the absolute most critical rule for our factory. Because of this highly specific structure, our enzymes are strictly one-way operators. They are literally only allowed to build a new strand in the 5' to 3' direction. No exceptions. So what this means is that when the copying actually happens, the original double helix pulls apart, and each of those original strands steps up to act as a template. The end result? Every single new DNA double helix is exactly half old and half completely new. You'll know this as the semi-conservative model of replication. It's this incredibly elegant mechanism that Watson and Crick predicted and was later beautifully proven by the experiments of Meselson and Stahl. Moving on to section 2, unwinding the double helix. Time to open the factory doors. Let me introduce you to our very first factory worker, helicase. Think of helicase as the ultimate molecular unzipper. It basically just barrels right along the DNA, smashing through those hydrogen bonds that hold the nitrogenous bases together. By forcibly untwisting that double helix and separating the parental strands, helicase is essentially prying open the factory doors so the rest of our machinery can get inside and actually get to work. But we run into a little snag almost immediately. Those newly separated DNA strands? They desperately want to snap right back together because of their chemical affinity. Well, here's where some awesome molecular teamwork comes into play. Right behind helicase, these things called single-strand binding proteins, or SSPs, jump right in. They bind tightly to those unpaired strands and act like microscopic doorstops. They literally just stabilize that open fork and keep everything perfectly separated so work can continue. Now, if you've ever tried pulling apart a twisted rope, you know the unseparated part gets tighter and tighter until it just bunches up into a knot. DNA does the exact same thing. So to prevent the DNA from getting too tangled or even snapping ahead of the replication fork, we have topoisomerase. This enzyme is the tension reliever of our microscopic factory. It carefully breaks, swivels, and then rejoins the DNA strands just ahead of the fork, safely relieving all of that dangerous overwinding strain. All right, section 3, building the leading strand. This is where we see some smooth, continuous assembly. Okay, so our main builder, DNA polymerase, actually has a pretty major flaw. It cannot start a brand new strand from scratch. It just can't. It needs an existing chain to hook onto. So an enzyme called primase has to come in first to save the day. Primase uses the parental DNA as a template to drop down a super quick 5 to 10 nucleotide long RNA primer. That little RNA segment provides the vital starting block that our assembly line desperately needs to get moving. Once that primer is finally in place, the real star of the show arrives on site, DNA polymerase III. This enzyme latches onto that RNA primer and just takes off, rapidly adding complementary DNA nucleotides directly to the free 3' end. And because this replication fork is opening up in the exact same direction that DNA polymerase III is traveling, it just keeps sliding forward. It builds this newly synthesized leading strand continuously right into the widening fork. Easy peasy. But wait a second, let's pause and think about the geometry of this whole setup for a minute. If DNA polymerase can only add nucleotides to a 3' end, meaning it's physically forced to move in a 5' to 3' direction, what in the world happens on the opposite template strand? Remember, that strand is anti-parallel. It's pointing away from the opening fork. How does that work? Well, that brings us to section 4, assembling the lagging strand. This is all about discontinuous construction. This part right here is notoriously tricky for students to visualize, so stick with me. Because of that strict one-way rule we talked about earlier, the factory actually has to work backwards, away from the fork, in these really frustrating little chunks. So instead of that smooth, continuous line we just saw on the leading strand, this lagging strand is built discontinuously. Basically, the fork opens a little bit, and a short segment is built backwards. Then the fork opens a bit more, and another short segment is built backwards. These clunky stop-and-go pieces are called Okazaki fragments, named after the scientists who discovered them, of course. To give you a sense of scale, in E. coli, these fragments are roughly 1 to 2,000 nucleotides long. Let's walk through how this lagging strand comes together step by step, because it requires some absolutely intense enzyme coordination. First up, primase has to constantly lay down new RNA primers for every single fragment. It is working overtime. Then, DNA polymerase III swoops in and builds the Okazaki fragment until it physically bumps into the previous primer. Next, a brand new worker enters the floor, DNA polymerase I. Its entire job is cleanup. It replaces those temporary RNA primers with actual permanent DNA nucleotides. And finally, we're left with these tiny little gaps between the backbones of the fragments. So DNA ligase comes in acting like molecular glue, and it seals the sugar phosphate backbone together into one continuous solid strand. Just incredible teamwork. Look at the coordination here. I highly recommend you just take a screenshot of this right now for your notes. This is literally the ultimate roster of our cellular factory workers and exactly what their jobs are. From helicase busting things open at the front, all the way down to ligase gluing things shut at the back. I'm telling you, mastering what each of these 7 proteins does is the absolute key to acing any exam on DNA replication. And finally, Section 5, Proofreading and DNA Repair. Let's talk quality control. To truly appreciate the sheer scale of what's happening inside a cell, you have to understand the speed. Get this. In bacteria, DNA polymerase III operates at a staggering, mind-bending rate of 500 nucleotides added per second. That is incredibly, almost incomprehensibly fast for a biochemical reaction that requires such high-level precision. Yet, despite that absolutely blinding speed, the final error rate in a completed DNA molecule is a minuscule one in 10 billion nucleotides. Seriously, how is that even possible? Well, it turns out DNA polymerases actually proofread their own work in real time. The very split-second an incorrectly paired nucleotide is added, the polymerase recognizes the mistake, deletes the wrong letter, and slots in the correct one before moving on. It's almost like having a backspace key that just presses itself. But sometimes damage happens after the replication process is completely finished. Like, say, when UV light from the sun distorts the DNA in your skin cells by creating these things called thymine dimers. When that kind of structural damage occurs, a highly specialized repair team initiates something called nucleotide excision repair. A nuclease enzyme acts kind of like a tiny mechanic, cutting out the damaged sequence. Then our old friends DNA polymerase and DNA ligase jump right back in to fill the gap and seal the repairs, simply using the undamaged opposite strand as a perfect guide. Which leaves us with a truly fascinating thought to wrap up on. As incredibly flawless as this biological machinery tries to be, constantly proofreading and repairing itself, it isn't absolutely perfect. An impossibly rare, uncorrected mistake does occasionally slip through the cracks to become a permanent mutation. But think about it. If this microscopic molecular factory had an error rate of literally 0, the genetic code would just never, ever change. So does that mean the spectacular diversity of all life on Earth is actually entirely dependent on the fact that this factory is just a tiny, tiny fraction of a percent imperfect? Definitely keep that in mind as you review your notes. Thanks so much for joining me on this explainer, and happy studying!